Hello again, back with membrane proteins. Last time I talked about how difficult it can be to obtain membrane proteins to do structural studies using X-ray crystallography. So I thought I would just show you really quickly another approach that can be used to make predictions about proteins which are possibly involved in membrane functions. So if you isolated a new gene that you were interested in, and didn't know anything about its structure, you couldn't purify it, you didn't really know what it did, you can subject it to what's called a hydropathy analysis. And so you can infer whether a protein is possibly a transmembrane protein by looking at windows of amino acids along the length of the polypeptide chain, and then determining whether the window is predominantly hydrophobic or predominantly hydrophilic, and that comes from the characteristic of the R group. So this is done by a computer algorithmically, and so when you look at these windows, you just slide the window along, and the computer algorithm looks to see, is this stretch hydrophobic, is this, and then it uses that to determine whether there's a high probability of that stretch of amino acids forming an alpha helix, which would then be a hydrophobic transmembrane region. So here's a hydropathy plot for a protein called connexin, and you can see that there in this part, in the first 50 amino acids of the connexin protein, there is a window of amino acids here, probably 20 or 30 amino acids long, which has a high hydropathy index, and that means that that stretch is relatively hydrophobic. That means it would be more thermodynamically favorable for those amino acids to be arranged into a helix embedded in a membrane. And then you can see the hydropathy index drop back down for this window. There's another window here where the hydropathy index goes up, and again here, and again here. So what this tells us is that it looks like that in this protein there are potentially four transmembrane regions. Now this is done on connexin, which we know is a 4-pass transmembrane protein with an amino terminus on the inside and a carboxy terminus inside of the cell. So when you have a protein, you don't maybe know whether it's transmembrane or membrane associated, you can do this hydropathy analysis. And so that's just kind of a bit of a shortcut, so that you don't necessarily have to do a full-on crystallographic analysis to determine the structure of a protein. You can get some structural information from the primary sequence and the relative hydrophobicity of different sets of amino acids. So that's it. I'll be back soon.